All right, I hope everyone can see. And thank you so much for your kind invitation. And like I said, I always love to talk about myself. So, and chess is, I fear sometimes it's lost in the noise of all the big laboratories, the big DOE laboratories. And so I thought I'd just stop for a moment before I get into the SAXS part of the lecture to give you the big picture. As a synchrotron, we've been around quite a long time, and we just completed in 2019 CHESS-U, right? A major upgrade in which I'm showing the part of the ring that sticks out of the hill, right? Most of our ring is underground. And on your right here is sort of a large gymnasium-shaped experimental floor, which we completely emptied out. It was spectacular to see this completely empty and built from the ground up new beamlines in this area. Fortunately for us, and I'll be telling you about my beamline, which is HP BioSAXS out here in Sector 7, did not get very much rebuilding or changes to it. So we were actually the first beamline in the facility to see new light. And it's amazing, it still impresses me that after completely dismantling this whole sector of the synchrotron and reassembling it, that they turned the thing on and the light shows up right at my undulator, kaboom, and we got a fluorescent spot. So it's like a moon launch, hitting a little tiny target from far away. So it's really a tribute to the engineers and scientists who make the ring run. So you'll also see, I'm just going to mention briefly, and I'll talk about just a little bit about these other facilities as it relates to SAXS and biology. You'll notice there's a hexagon here, HMF, that's high magnetic field. We have been awarded a significant grant to build a new building containing a state-of-the-art high magnetic field. And so for those of you who may be familiar with our brown building over here on the right, we're basically demolishing all the trailers and stuff and building a special building to house a state-of-the-art magnet with with a very large cavity so that you can shine X-rays through it and put samples in the state-of-the-art magnetic field. Now, this is primarily going to be quantum materials and material science kind of applications, but it initially will be a SAXS configuration. And there was some biology written in the proposal. So if you can think of interesting things that combine SAXS and very intense magnetic fields, please let us know. There will be opportunities to do stuff like that. So I just thought I'd mention it because that's kind of the most exciting thing that's happening. And so actually this building, they are breaking ground on it imminently. And so this summer will be a lot of digging and jackhammering and unfortunately rather close to my beamline. But the remarkable thing is that the plan is that we will not lose any actual beam time as a result of the construction. So operations will continue. They've scooted things around. We have longer breaks than normal, a little more crunched in some places, but miraculously not losing any time. So that time is 125 days a year for partners. And we have a partner model now, and that's 24 hours a day. We go down on Tuesdays for maintenance and so forth. And then there's some additional time at the director's discretion. And currently we're NSF, both DMR, bio, and engineering divisions support us, and particularly in Sector 7, we're NIH and State of New York as well. So I just wanted to put a couple of points before I move on to SAXS about what I think makes us different and unique. We just finished this CHESS-U, so we have new beamlines, a new experiment floor, except for me. Major developer of detectors and undulator technology, as I'm sure many of you have heard that those are big efforts here. We are located on a university campus, and we think of ourselves as the nation's teaching synchrotron. So you will see, I'll give you some examples of all the student participation that goes on. Easy access, right? There's no gate. We're not a government facility that has to be careful about that kind of access. We're a university, and we have to be open to the nation's, the world's researchers. So that's easy to get on campus. we have this new partner model. So in other words, the NSF is no longer the steward of this facility. We have partners who share and pay for the operation of the machine. The NSF is still a big stakeholder, but that gives us some flexibility that we don't necessarily have to have all fundamental science we can have directed goals. And for example, the Air Force is a big player right now, and they have 2 beamlines. So, and they work rather differently than traditional NSF. We have greenfield expansion capability. That's sort of lingo for, we've got beamlines to rent, so, or build. We have space to expand. And then the usual things which I've always been proud of are our tightly integrated technical staff and so forth. So I mentioned there were other beamlines. Now we're HP Bio over in Sector 7, and 7B is crystallography, which I won't really talk about. We have then PIPOX, which is spectroscopy, and actually a lot of biology gets done over there. That's X-ray spectroscopy. And then quantum materials and shaping and forming, and then the Air Force runs functional materials and structural materials. So that's a soft matter beamline and a high energy hard matter beamline. Okay, so that's basically what's going on right now at CHESS. So I'm going to talk about Sector 7A, right? So our mission is fundamental biology and biomedicine, and biomolecular structure interactions. High pressure is something that is uniquely offered, and I'll talk a little bit about that momentarily. So I just wanted to introduce you to the people at Sector 7. So Qinshu Huang and I basically run the beamline, and we share responsibilities. Qinshu is a very capable molecular biologist who can run the station without me. So a very invaluable person, and you will no doubt, if you come, interact with him as well as me. We have graduate students. Robert Miller is a half-time graduate student whom I share with Nozomi Ando's group, and I'll show you some of his work. Gabrielle Ilova has worked with us for a number of years. She started as a community college student through a program, switched to Cornell, got honors in physics, went on to graduate school, and is sort of in the final years of her thesis work. So I'll show you some of that stuff. Ben Myers is doing some simulation for me, and we have the usual characters who we rely upon heavily for advice. I just want to point out that we can do high pressure crystallography, and Zhongwu Wang, who's a diamond anvil cell expert, heads up that endeavor. And by the way, Aaron Finke, who has been with us for a number of years and really brought the serial crystallography program to fruition, has moved on to back to Europe, where I think he really wanted to go. And so we're currently searching for a staff scientist in that category. So we have some positions open. Just for the beamline jockeys who might be listening, we have an undulator, Cornell compact undulator. We have a white beam mirror, and then we have a multi-layer monochromator pair. The bandwidth there is about 1.5%. And then we have some vertical focusing, and that's basically the beam coming into the hutch. We can do between 4 and 7, almost 15 keV at the maximum. And our beam is usually 250 microns square-ish, but we do have some focusing optics, which can get us down to 50 to 30 with some gain, right? And so flux, typically right now, we're running the ring at 100 milliamps and at 10 keV, and we typically are sort of in the mid 10 to the 12 range in terms of photons in the spot. And we're expecting when we finally ramp all the way up to 200 milliamps, which will happen this year, I think, we should be in the 10 to the 13 range at that point. Okay, and so for Q space, we get, it's typical SAXS station. We can get down, if we really push things, to 0.005 or 0.006 inverse angstroms, and we can get up beyond 1.0 in terms of wide angle. That's user selectable. So we have an Eiger 4M that's in a vacuum tank on a track, and the user can move it back and forth to where they want it to be. And then, of course, at 14 keV, we can get to we take off the front and run an air, we can get to quite wide angles there. All right. So good, only 10 minutes. I'm not going to waste my time with percentages. If you want to know, you can ask us. Here's sort of a timeline, just to let you know. We had first light in the end of 2019. We installed the Eiger 4M in the vacuum. That was an interesting, that whole thing was made in-house, by the way. We have welders and so forth who know their high vacuum stuff. So that was kind of fun. You can see that's Robert's, my graduate student, Robert, sticking his head into it. So it was a rare opportunity for a graduate student to build new stuff like that, and got out to some of our records. I'll tell you about our fancy high pressure stuff, which I'm very excited about. We also, Gabriel Ilova has designed an anaerobic chamber that rolls up to the SAXS station. So that also, I think, is kind of unique, and I'll tell you more about that as well. I should point out that during the shutdown, Chess made a very big effort to go totally remote. And we did, thanks to Elke Ehrenholz, who was our leadership here at the time, really pushed and drove us to achieve, I think, some really wonderful ability that chess-wide, we were able to hit the ground running and run remotely for users. So I'll tell you a little bit. We continue to have that capability, and I'll tell you something about that later. So again, during that period, I just want to give you a sense of, we are sort of half funded by NSF and NIH. And so in blue are the conventional BioSAXS users. There's sort of, most of them are NIH biomedical applications, and in yellow are our high pressure SAXS. So we're building a high pressure biology community here, which I'm very excited about. I think it's a very exciting story, a novel technique. And here at the end, we were finally allowed in 2020, these are the 3 running periods per year. So last fall, we were finally allowed to have in-person groups do setups of special equipment. That's what's red. So they take a lot of time to do, but they're very important as well. So we do a mixture of standard SAXS and exotic stuff of different kinds. Yeah, so anyhow, the tank is fairly straightforward. Like I said, it's just a giant coffin with a sled in it that the detector rides on. And we have a gate valve now, so that if you need to break vacuum, just shut the gate valve and swap out your, this blue box is the standard SAXS setup. It's got a camera and a capillary flow cell through it, but you can shut the gate valve and swap out all kinds of equipment without having to disturb the detector vacuum. So that's proven to be very, very efficient. Yeah, and so here's actually a picture through the, there's a remote pan zoom camera. We had cameras installed everywhere at Chess. So you can watch live what's going on in the hutch. I don't quite have the audio set up so you can yell at people, but I'm working on that. But anyhow, this is, so the solo robot is a high throughput, 96 well tray for loading samples. There's a little, you can't really see it here. I had to bring this up live actually. There's a little funnel that it loads samples into that then go into the capillary, pumped into the capillary. And alternatively, we have a valve where we can, with the push of a button, switch to inline size exclusion, which is very popular. At least half of our users are doing the size exclusion instead of or along with the regular type of SAXS. So that's our normal SAXS setup. We do have dynamic light scattering. I'm not nearly as proficient with it or heavy user as the BioCat guys are. They really take a lot of advantage of that, but we do have it, and it is available. So this is the interface, which actually was developed by SAXS Lab. So Soren Sku, who is a scientist at SAXS Lab in charge of the biological scattering, developed, worked with me for a couple of years here when he was postdoc, and graciously has allowed us to piggyback off of their GUI for collecting samples, which we have modified to work on a beamline. But if you have a SAXS Lab instrument, this will be immediately familiar. And in fact, we have a SAXS Lab instrument on campus. So we think that ease of going back and forth between, we think lab sources are a good thing for synchrotrons. They help, help people do better, more successful experiments. So I heavily encourage that. And we love the idea that we're compatible. If you teach someone on a lab source, they come here and they can see, it looks all familiar. All right. So also, one of the nice things about having students is if you really want something that's easy to use for users, you put an undergraduate on to see if an undergraduate can do it who's never picked up a pipette. So Ben, who's a superb physicist, so had him be the first person to do online remote. This was before undergraduates were allowed in the facility, and he was actually the first one allowed back under COVID conditions. But before that, had him do some remote data collection just so that we could. And then we put on our first group. This was Emily Weinert's group at Penn State. And thanks to Neela Yenawar, who's a longtime friend and colleague, Yenawar's friend and colleagues, who has brought people from Penn State to work with us for many years. And this is Dana Pat. So we had Chin Chu loading samples in the facility. And Dana Patterson, this is her baby. This is her project. She's a grad student for Emily. And so they were able to, they had this hypothesis about this is a gas sensing protein of some kind where domains change their states. And we collected data, and I walked them through the processing. The students, had never done this before. And lo and behold, ta-da, it was a great teaching moment. We could see this dramatic change in the structure right away with the processing. I could show them how to calculate this stuff using the BioSAXS raw, which again was Soren's original project, and we've developed it here for more than a decade. Now Jesse Hopkins at BioCat is the steward and the big developer of that. So I have happily turned this baby over to him, and we continue to benefit from that relationship. So anyhow, Dana's and Emily's project just came out as a Proceedings of the National Academy of Science paper. Very nice stuff. And so that was our very first COVID remote data collection. So we continue to be able to do that. We also support other exotic stuff. So here, I just thought I'd pop up. We did, the Ando group has been working, as I said, on anaerobic SAXS, which I'll show you examples of that. And so they're interested in sophisticated spectroscopy in real time because they're monitoring these, these iron sulfur complexes and other exotic things that are ferociously oxygen sensitive. And so it's a very good test. We have, the Nagel group did, this is sort of like grazing incidence SAXS, only not quite. They're looking at membranes deposited on surfaces. They shipped us a box of equipment. I think they were quite skeptical that we'd pull this off, but we did. We got their system running completely while they talked us through it on Zoom, and I think got them better results than they've ever had before. So that was a great success story. There are two manuscripts that are now in preparation on this, looking at small peptides that are being designed to help replace antibiotic resistance, help battle antibiotic resistance. And they're looking at how the peptide insertions change the ripples on the membrane surface. So also down here, we did, we also were able to do our high pressure experiments remotely. This is high pressure chromatography that was being done with the Royer group. I'll talk about that in a moment. So this is my reminder to go live and show you how to get time, how to figure out when the beamline is operating. So I'm going to see if I can do that. Whoops, okay. Let me pop up my, where's my, there it is. Okay, so here's the important website that you all should know about, right? www.chess.cornell.edu, all right? And that'll bring you to this website. And this is where there'll be news and so forth. And you want to go to users, and you'll see that under users, there's a user guide. for prospective users, user guide, and then I'm just going to go, and so those are important things to check. I'm going to go straight to the user portal because that's how you register in our database and request and submit a proposal. Okay, so we go in here. Now, as a first-time user, you would hit register and you type in your email address and some other information, and it will give you an account. Now, I've obviously already done this, so I won't go through the whole process, but I will just give you a taste for how this works. How I hope it's easy. It's designed to be easy. If you remember your password, hang on. All right, there we go. And that brings you into the user portal. I hope everyone can see this, right? Okay, so in the user portal, you will see users. Oh, and you'll see proposals. And under proposals, you want to scroll down and go My Proposals. Okay, and that lists all the proposals as just tests here. And so you go New Proposal. You got that? Okay, and we'll call this an APS test setup, just to confuse the user group, user office. Okay, and principal investigator is me. Okay, and I am affiliated. They'll want to know who you're affiliated with, and run estimated 8-hour shifts, right? So it's a shift is 12 hours. You know, if you want a day, that's 3 shifts. See, contact me if beam time becomes available. For biological stuff, we have a rapid access. You just put the words rapid access in the title, and we will consider you as soon as possible and try to squeeze you in. So we can do that. And the turnover is pretty quick. It still has to get reviewed. Run cycle, right? So it's 2022-1 is the one that we are, see, rapid access submission only, the one that's underway right now. And there's 2022-2 and 2022-3. So we have 3 of those, total of 125 days. Those are 8-week runs, usually with a break in the middle. One in the, right in the spring, right now, starting at the end of January. And then another one that lasts until, normally until July. We're cutting it short. We're adjust shifting times around this run. And then we have one in the fall. So I'll say this, and then you can request the dates and so forth. Requested dates, test, okay, testing. All right. So now you want to add investigators. I'm going to say there's no collaborators. You want to add all your investigators. All right, and then sources of funding. And then eventually, I'm just going to go down scientific justification, right? You have to, this does go out for review, peer review, and it gets ranked. So you do want to put in justification why it's important science, evidence that your group knows something about SAXS and it's an appropriate place to do the experiment and so forth. All right. And so once you've done all that, then you hit your, submit. And then those will go out for review. Our scheduling person will contact you and work with you to get into the schedule. All right. So I hope that all makes sense. Let me just pop to the main chess. I'm going to get out of this right now, log out and go back to the main chess website again. Just so that you know what the schedule is, and it gets announced all different places. If you look at the X-ray run schedule, that will tell you when we're running. So I'll pop this up, and right here is the, here's where we are right now. You can see, so the light blue is alignment period. You're getting the stations ready for users. And then on the Wednesday, next week, the deep blue is our 8 weeks of user running. It's a little unusual this year. So that's where you find out what the schedule is. Okay, great. Okay, I'm going to head back to my normal presentation and finish this off. Okay, let me just pop back again. I'm getting ahead of myself here. All right, yeah. So Oh, my timer's reset. Looks like I have maybe about 15 minutes or something. Okay. So yeah, so anyhow, yeah, this is a cool story. Gabrielle, like I said, was a community college student. And anyhow, she developed we basically had a custom designed koi anaerobic chamber, which wheels up to the station and has all of the connections so that you can prepare your samples. You have everything you need in this box to do really very sensitive oxygen compounds. You get the centrifuge and all of that business. And then you can either do chromatography or batch oscillating, flow cell SAXS directly there. And so she has a manuscript in progress, and then this is open for other people to use in future runs. We actually do have one external user this run. All right. Now, I just want to talk to you a little bit about the more exotic stuff that we're doing to finish up here. Now, you've heard that we are a high pressure biology facility, and for years I have helped Gruner Group do these weird high pressure experiments and didn't really think much of it. And then one day, he was making the case, well, we should fund a beamline to do this. And I was skeptical. I started reading and I was blown away. I didn't realize how much this field has come of age and how relevant it is to what's going on in biology today. And so it has been used, high pressure has been used in biophysics for some time to understand folding and protein-protein interactions and catalysis. But what has happened more recently that has changed the landscape is that food science uses high pressure to process food. And this is now a multi-billion-dollar industry. You go to the grocery store and get all kinds of things come in these little pouches that don't need refrigeration. And what they're doing is they're pressurizing those babies to 600 or 700 megapascals. That's 6,000 or 7,000 atmospheres. Kills the pathogens, and it preserves. You don't have to cook it or add dangerous chemicals. It preserves the flavor and the nutrition, and it's a big deal. So people in that field are interested in how does pressure change the properties of biomolecules? How does it? What is the limits of life? And why is it that some microbes, such as archaea, can survive at pressure, and others, such as eukaryotes, cannot? So, but also most interesting to me personally is the deep life. That is the increasing realization that the biomass on this planet is not what we see up here on the surface. It is stuff that is under the water and underground. Most life on this planet is under some kind of pressure and extreme conditions. And so there are big and interesting questions about that, which I could go on and on. I'm not going to waste our time here, but so the ocean bottom is about 1,000 atmospheres, right? It's 109, 110 megapascals. It's conceivable in some of the subduction zones that life might be up to 300 or 400 megapascals. And they're able to pressurize. Some microbes clearly can adapt to gigapascals if you survive them for short periods of time. But this is so hot and deep are extreme conditions. We have 4 technologies. The 2 of them that are SAXS are the standard high pressure SAXS cell, which is static non-flowing SAXS. And then the more exotic high pressure SEC-SAXS, where we can do chromatography not only under pressure, but we also do the detection under pressure. So we can keep everything under pressure. And here's a picture. So it's not much different than the regular facility. We put a box here. We can heat this thing up and cool it down from 0 to about 80 degrees C. So we can do thermophiles, deep ocean hydrothermal vent kinds of conditions for life, as well as cold denaturation. So interesting extreme conditions. There's the paper where Durgesh Rai, who was postdoc here, who's now in the SAXS lab, Xenox, was the author on that. And this is this is Saul Gruner. Actually, Nozomi Ando's original research. So it goes back, but this is, yeah. So that's what it looks like. It's a big block of metal. You load stuff into this, easy to load mechanism, and you shine X-rays through the diamond windows. It has to be a high energy experiment because you get absorption, a lot of absorption from these big, thick windows. So anyhow, that's the idea. And so why is it? I just give you a few examples of the cool things that happen, right? So the simplest thing, the first thing that happens is, biomolecules have to minimize their volume when you pressurize them. They try to, you're biasing for minimum volume. And surprisingly, what does that mean? It means dissociating and unfolding. Why? Because biomolecules are never perfectly packed. And the only way that they can really decrease their volume is unfolding and coming apart. So it's a very interesting, and it's almost counterintuitive. So you can use it as a way to, in a controlled fashion, dissociate complexes and see what changes. And then if you really put the pressure on, you get unfolding. And so that's what the biophysicists like Kathy Royer are interested in as a tool. All right. And so we have some papers out on that. I won't get into details. Here's the cool stuff. All right. So it's not just proteins that do interesting things. Lipids do even more interesting things, or even more sensitive. And all living things depend on lipids, I think. I'm pretty sure, as far as we know. So this is a group, you may have seen them in the news, right? This is a group at the Monterey Bay Aquarium in collaboration with UCSD. It's Itai Budin and Jacob Wittikoff. So they have gone, on their way out, you may have seen in the news several things of excitement. They saw the transparent head fish, I think, is something they observed that was all over the news. And the mammoth tusk, they were searching, miles and miles out to sea at under 10,000 feet. And it's like, oh, what's that? That looks like a mammoth tusk. And it was like, hundreds of thousands, or, ancient tusks. So anyhow, they're collecting jellyfish from different depths in the ocean and looking at the lipids, lipidomics, and are interested in why on earth do jellyfish change their lipidomics to adapt for pressure. This is sort of a rules of life kind of thing. So, and what's nice is just under fairly reasonable pressures. I mean, these things you don't have to go all the way to the bottom of the Mariana Trench to see these changes. So, and all of it, from both live samples and increasingly, they're starting to figure this out. They're running simulations and understanding the connection between, this is how an organism, this is phenotype and genotype and the basic physics. So how the organism utilizes the basic physics, what rules of life when it's under pressure. So this to me is really exciting stuff. And so these guys brought jellyfish goop. First, they sent it to us by remote because it was still COVID, and then they got to visit us most recently. And so they're in the process of doing that. So the final cool technology I just want to show you is this Robert Miller, who is in the middle of doing his PhD in the Ando group over in chemistry, works half-time with me. And we're working on a manuscript together where we have made a chromatograph that can go up to deep Mariana Trench-like ocean pressures. And it has a special hardened capillary cell that can withstand the pressure at single crystal sapphire. And it's controlled so the pressure is uniform. And we can do the temperatures anywhere from, cold, 3 or 4 degrees C, like most of the abyssal plains of the ocean, to the hydrothermal vent temperatures like 50, 60, and so forth. And one of the things, so just an example of what stuff looks like, this is a chromatography trace of PP32, which is a peptide that's used for protein folding studies. It does interesting things under pressure. And you can see the radius of gyration, it changes under pressure. And if you look at it, what it's doing is unfolding. This is a Kratky plot, and the wide angle part of the Kratky starts to go up. And that's characteristic of an unfolding. And then you calculate the P of R function, and you can see the thing is getting bigger, right? So simple, you don't really need a chromatograph, but you can see those things happen. Far more interesting situation are proteins that dissociate. This one is yeast enolase, And if you at room pressure, right, it's entirely a dimer, room pressure and cold, where this is cold dissociation we're looking at 4 degrees C. Then you put the thing under pressure and the peak looks the same, but underneath the peak is now a mixture of monomers and dimers, which rapidly equilibrate, all right? So, a more interesting case is L-lactate dehydrogenase from wild boar. And there, at room pressure, you actually see a single peak, which is entirely tetramer. And then as you pressurize it, you see a second peak develop, which is predominantly dimer. And you can separate those under pressure and get structural information because this is a case where the exchange between, once a dimer breaks off from a tetramer, there's a little conformational change. And so the recombining is not quite so easy. So that's called conformational drift. So I just wanted to give you an idea of sort of the cool things that are going on. We run workshops. We did our first high pressure training workshop. It was entirely online. We are planning an in-person one this April. So if you are interested in learning high pressure BioSAXS, this is an NSF funded one that is coming up in April. So check out our website. We're going to have 7 and 8 will be world-class researchers. Hopefully some of our international people can get here. We're gambling that Omicron will have passed by then. So we have a nice lineup of speakers on the 7th and the 8th. And then Saturday through the weekend, we're doing a hands-on teaching people how to run it in person. We do damn too many workshops, if you ask me. This was extreme conditions. So anyhow, yeah. So if you want to ask me about other stuff, I'm more than happy to quit here and open up for discussion.